Okay, we're looking at chapter muscles. So, looking at the anatomy, you have a muscle to bone attachment, which is connective tissue. And if it's a rope-like connection, they refer to it as a tendon. The outermost connective tissue surrounding the whole muscle itself, you can see here, is referred to as epimysium. Epi means upon. So, all upon circling the muscle is called the epimysium. Now, if you were to pull out one of these units out of the whole muscle, and you can see it in this picture down here, it's referred to as a fascicle. So it's all this structure right here. And the connective tissue surrounding the fascicle is referred to as the perimysium. There it is. And then if you pull one of the units out of the fascicle, now you're at the cell level. You can refer to it as muscle cell or muscle fiber. Fiber indicates that it's very long. And then the actual connective tissue around the muscle cell itself is referred to as the endomysium. And endo means within. So starting again from outside most, you have the connective tissue around the whole muscle called the epimysium. The unit inside the muscles, pull one out, it's called a fascicle. Connective tissue around the fascicle is referred to as perimysium. And then the units within the fascicle, pull one of these units out, it's a muscle cell, or you could refer to it as muscle fiber. And the connective tissue around the muscle cell is referred to as endomysium. Now that we've kind of gone over some terms with the wording myo is referring to muscle. Okay, let me go to the next slide here. So we're going to look at a muscle cell, and kind of just to reiterate what we just mentioned, the connective tissue around the muscle, which is not labeled here, is referred to as endomysium. What you're looking at here is the cell membrane. The muscle cell gets a fancy name for the cell membrane. It's referred to as sarcolemma. And if you can remember that skeletal muscle, one of the features about this muscle is that it has striations where you see dark regions and light regions. And the dark region or striation is referred to as the A band, and the lighter region is referred to as the I band. And you can see at the cell level, nuclei. And if you remember, skeletal muscle has many nuclei because when you develop as an embryo, they fuse all together to form one long skeletal muscle cell. Pull one of the units out of the muscle cell, you're going to have what's referred to as the myofibril. Okay, let me clear all that. So we're actually looking here at a slide representation of the skeletal muscle where you see the striations. The dark regions are referred to as the A band. The light regions are referred to as the I band. You don't even actually get to see the whole muscle. It's very long. So muscle's going across this way, and then you have another one right above it going across. And again, a fancy name for muscle cell. You can refer to it as muscle fiber as well. Pull one of the units out of the muscle cells, referred to as a myofibril. And we're looking at one myofibril. So it does have the striations, again, where the dark regions are. It's referred to as the A band. And the light regions, where it looks lighter, are referred to as the I band. So basically, you have proteins inside the muscle cell. When you get down to this level, you're at a myofibril. So the proteins are referred to as myofilaments. There's a thick myofilament. The name of the thick one is called myosin, and it's here in this brownish red coloration. And then there's a thin myofilament in blue, referred to as actin. So all this in blue is the actin. The functional unit, which is the smallest component of a muscle cell that does its job, is referred to as a sarcomere. And it is a region from, we say, one Z disc to another Z disc. And within the middle of the Z disc, you have a zone referred to as the H zone. And typically when muscles get excited, they will contract. And when they contract, they get shorter in length. So as the muscle contracts, this sarcomere is going to get shorter in length, and you're going to see these Z discs being pulled closer to the middle here of the sarcomere. Okay, we're looking at inside the muscle cell, you have myofibrils, and you have this organelle in blue, referred to as the sarcoplasmic reticulum. And inside the sarcoplasmic reticulum is calcium that is stored in order for the muscle to contract. So when the muscle gets excited, calcium is going to be released, and that's going to aid in the muscle to contract. And calcium is going to be released in an area referred to as the terminal cisterna of the sarcoplasmic reticulum. So if I have a look here, the whole organelle itself containing calcium is called the sarcoplasmic reticulum. Within the middle here, this is referred to as transverse tubule. It's basically an infolding of the cell membrane. And then on two sides of the transverse tubule, it's referred to as the terminal cisterna. And that's the area where the calcium is going to be released. All right, so here you're looking at the top picture where you have the actual slide of the skeletal muscle with striations. You're looking at the cell level in the middle, where the cell membrane is referred to as the sarcolemma. Inside are units referred to as myofibrils. Within the myofibrils, or even within the muscle cell, you're going to see striations where you got a dark band. It's referred to as the A band, and the lighter striation is referred to as the I band. And what causes those striations? Let me see if I could just back up a little bit. Oops. What causes the striations are the proteins myosin and actin. And I stopped sharing for a period of time. Let me try to get that back real quick. Okay, going back to what causes these striations are the proteins, the myofilaments. So the thin one is called myosin, and there's a thin one in blue called actin. If they overlap, it's going to look darker. If you only see the actin, it's going to look lighter. So that gives us the I band. All right, let's go ahead and let's go from the myofibril in the middle picture and magnify the functional unit referred to as a sarcomere. So basically, what you have here are the thick proteins myosin, and they have these little head regions, and just like hinges, like your elbow has a hinge, is able to attach to actin and move it with the hinges. So these myosins on the right side will be moving the actins toward the middle, which is referred to as the M line. The myosin heads on the left side have hinges that once they bind the actin are going to move the actin from the left to the right. And when a muscle gets excited, again, it gets shorter in length. So therefore, the sarcomere will be getting shorter in length. And that's referred to as the sliding filament theory. Now we look at the myosin heads. You have an area where it does bind to ATP. That's that molecule all cells need to produce in order to do work. So that ATP is going to provide the energy to move this hinge so that once myosin binds to actin, it's going to move it toward the middle of the sarcomere. Also on the myosin head, you have some areas where it's going to bind to actin, which would be right above the myosin heads. When myosin does bind to actin, it's referred to as a cross bridge. So here's the actin, and before muscle contracts, the binding sites where the myosin likes to bind to are covered up. So they're covered up by a protein referred to as tropomyosin. In order to get those binding sites exposed so that myosin can bind to the actin, you need calcium. So when calcium is released from the organelle called the sarcoplasmic reticulum, the calcium, let's say it's this little green spot here, it's calcium, it's going to bind to a protein called troponin. And when calcium binds to troponin, it's going to push aside the tropomyosin and therefore expose these binding sites on actin. And that's how the calcium is used in a skeletal muscle contraction. So you're looking at the top picture of a sarcomere, which is the functional unit of the skeletal muscle, which basically means the smallest component. It does its overall job. So when these myosin heads bind to actin and ATP is available, they can move the sarcomere on both sides to the middle and therefore be getting shorter in length. So again, you need calcium to expose the binding sites on the actin. Once that's done, myosin can then bind to actin. And the myosins on the left side here will move the actin toward the middle, toward the right, whereas the myosin heads on the right side bind to those actins above it, and they'll move actin from right to left. And therefore the sarcomere gets, again, I can repeat it, shorter in length. And as the sarcomere gets shorter in length, you can see it here in this drawing, it's being pulled toward the middle, referred to as the M line. Now, how do you get a muscle to contract? Well, your nervous system has to communicate to the skeletal muscle through neurons, which is a fancy name for a nerve cell. At the end of the neuron is a region referred to as the axon terminal. So an action potential will be traveling down the axon, which is the tail of a neuron, which will eventually cause the release of a neural transmitter, a chemical used to communicate to the skeletal muscle. This particular neurotransmitter that will excite skeletal muscle is referred to as acetylcholine. The acetylcholine is stored in little vesicles referred to as synaptic vesicles. There's always a little space between the neuron and the muscle cell, never physically touches. So this little space here is called the synaptic cleft. Once the neurotransmitter is released, like a key to a lock, it has to fit perfect to a receptor protein in the membrane of the skeletal muscle. So acetylcholine binds to what's referred to as chemically gated channels, indicating that's how they open up. They open up in response to a chemical, which in this case is the neurotransmitter acetylcholine. Once the channel, this chemically gated channel, opens because of the acetylcholine, positive ions for sodium for this particular channel will want to flow in. And as they flow in, they excite the skeletal muscle. The term is, we say it's being depolarized. Depolarized means you're, going above its resting membrane potential. Let me clear all these drawings and see if I can get to a kind of clear screen here. Neurons, which are the functional units of your nervous system, communicate to targets. The targets they can communicate to are skeletal muscle, even cardiac muscle, smooth muscle, or glands. The way neurons communicate to targets is they try to create what's called an action potential in the target when they're trying to excite it. When a cell's at rest, if I draw a cell, let's say I draw a circle representing the cell. If I count through a device called a voltmeter, the ions inside the cell and compare it to outside the cell, inside the cell, it's more electrically overall negative. That doesn't mean you don't have positive ions inside the cell, it just means when you compare it to the outside, it's more electrically negative. Where outside, it's more electrically positive. And again, that doesn't mean you don't have any negative ions outside the cell, it just means when you compare the inside to the outside, it's more electrically positive outside. So this difference when the cell's at rest inside is referred to as the cell's resting membrane potential. And most cells, the resting membrane potential, if I draw a little graph, if I draw a chart, a little graph here, inside the cell, the voltage inside compared to the outside, for most cells, it's about minus 70 millivolts. Okay, let me draw the cell again. Now, if I were to open up a channel, let's say for sodium to come in, sodium, the element sodium is Na, and the charge on it is plus. There's more sodium outside of the cell. So if I were to open up, let's say, a protein channel, positive ions would want to come in from high to low concentration. As positive ions for sodium come in, the inside voltage of the cells being changed, becoming less negative. When it becomes less negative, the term is referred to as depolarization. Now, if I want this muscle to contract, I want to try to create what's called an action potential. What's an action potential? An action potential is when you change the voltage inside from minus 70 millivolts to positive 30 millivolts. So I want to get the inside to positive 30 millivolts. So the first step is to open up channels referred to as chemically gated channels for sodium. So that way sodium comes in and the inside's becoming depolarized. You also have other sodium channels, but they open up in response to a change in the voltage. So some sodium channels will only open up when you change the voltage inside the cell to about minus 50 millivolts. Once you make the inside about minus 50 millivolts, then voltage-gated channels for sodium will open. And when these voltage-gated channels for sodium open, they all open. If one's next to another voltage-gated channel, it'll open. Similar in this picture here, you're looking at a voltage-gated channel for sodium up here in the top right. And when one opens, they all open, creating your action potential, which the definition of an action potential is a brief reversal of the inside voltage from minus 70 millivolts to positive 30 millivolts. So if I create an action potential in a skeletal muscle, the muscle will contract. Okay. Now, neurons can communicate to other targets like glands. If I create an action potential in a gland, well, the gland's not going to contract. It's going to release whatever it makes. Neurons can also try to excite other neurons. And when they do that, you create an action potential. But a neuron is not going to contract. It's going to, when it gets excited, release neurotransmitters. So the whole thing here is you got a neuron, you have the end of the neuron referred to as the axon terminal. When the neuron is excited, it's going to release neurotransmitters called acetylcholine. Acetylcholine will bind first to what's called chemically gated channels for sodium. And as the sodium comes in, the inside's becoming depolarized. Once I reach a certain threshold, a change of the voltage, about minus 50 millivolts, then voltage-gated channels for sodium will open, indicating they only open up when you reach a certain voltage. When one opens, they all open, creating what's called your action potential. A brief reversal of the inside from minus 70 to positive 30 millivolts. And at this point, if we're inside the skeletal muscle is releasing calcium, which is needed in order for the muscle to contract. And then there's also the muscle has to come back to rest. And the term for coming back to rest is referred to as repolarization. Let's try to erase some of this here. So coming back to rest, you want to return the inside voltage back to minus 70 millivolts. All right, so how are we going to do that? Well, go back to our pen scenario. So at 30 millivolts, you also have voltage-gated channels for potassium. That means they're only going to open up at 30 millivolts. So at 30 millivolts, voltage-gated channels for potassium will open. And because there's more potassium, which is K plus inside the cell, it wants to go out from high to low concentration. And as potassium leaves the cell, you're coming back to rest. And that term is referred to as repolarization. So you can see up here on the bottom right that after you create an action potential, that voltage-gated channels for potassium will open, bringing the cell back to rest, back toward minus 70 millivolts. Now, these channels are open up a little longer than you need to get back to minus 70, so you fall a little bit below rest, and that's called hyperpolarization. But finally, they do close, and then what's called your sodium-potassium pumps and what's referred to as leaky channels, channels that are just naturally open, will bring you back to minus 70 millivolts. All right, let's clear all this and let's try to re-emphasize this, looking at some videos, going over what an action potential is, what the resting memory potential is, so that way you can try to visualize it. So just give me a minute for that while I pause the recording to get to those videos.
The human brain alone contains about 100 billion.
Okay, let me make sure I'm sharing audio as well.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, so that's a brief explanation of the resting memory potential. What you want to get mainly out of it is that when you compare the inside of the cell to the outside, it's more electrically negative. It refers to as the resting memory potential. For most cells, it's about minus 70 millivolts. Okay, let me jump back and let's look at another video. Let's look at what an action potential is. Again, the general definition is you're trying to change the inside voltage from minus 70 to positive 30 millivolts. That's an action potential. And if you create an action potential in a neuron, the neuron is going to release a neurotransmitter. Whereas, let's say if you create an action potential in a muscle, the muscle is going to contract. Okay. So let me magnify this and play the video.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels.
Now, let me jump in real quick and kind of reiterate some things I had mentioned earlier. Get the marker out. Okay. Anything below threshold, let's say anything above minus 70 millivolts, it's called depolarization, right? the first type of channels that are going to open up are not voltage, chemically gated channels for sodium. Chemically gated channels. So let's say I have my neuron, which is a nerve cell, which I'm drawing horribly here. I'm drawing the tail, which is called the axon. And at the end is called the axon terminal, where you're going to release the neurotransmitter called acetylcholine onto the muscle. And the first type of channels that are going to open are the ones that bind to the acetylcholine, and those are going to be referred to as chemically gated channels for sodium. In other words, they're only going to allow sodium to come in. Yes, a little bit of potassium will leave, but more sodium comes in. So when the acetylcholine binds to this chemically gated channel for sodium, it will open. I'm trying to spell chemical. And because there's more sodium outside the cell, it wants to come in. And as positive ions for sodium come in, the inside's becoming less negative, which again is referred to as depolarization. I'm not going to spell it all out. So right about here in this area, type of channels that are open up are for sodium, but they're chemically gated. Once you reach a threshold, they say, in other words, once you change the inside voltage to a certain level, and the threshold here is about minus 55 millivolts. So once I reach about minus 55 millivolts, let me get the correct color out. This is our threshold here. The next type of channels that open up are called voltage gated channels for sodium. So what causes them to open is just they sense the inside. The inside's about minus 55 millivolts now. And when one opens, they all open. And when they open, they also will allow for sodium to come into the cell from the outside. And that creates your action potential. So now you're going from minus 55 millivolts, opening up these voltage gated channels for sodium, and now you're changing the inside voltage to positive 30 millivolts. Once you get to 30 millivolts, if this were a muscle, the muscle will be contracting. Calcium's released and muscle contracts. After an action potential, yes, you want the cell to come back to rest. The term for coming back to rest is called repolarization. And the type of channels that will open up next are for potassium. Let me erase this. And the potassium channels are voltage gated. In other words, they're going to open up when they sense a change in voltage around 30 millivolts. So let me draw again our cell. And so when you reach about 30 millivolts, these channels for potassium are voltage gated. So they're not going to open until they sense it's 30 millivolts inside. And once they open, since there's more potassium inside, it wants to go out. And as you're losing positive ions, you're coming back to rest, and coming back to rest is referred to as repolarization. Now, these channels for potassium are open up a little longer than you need. You do fall a little bit below rest, called hyperpolarization. They'll eventually will close, and called leaky channels, which are naturally always open for sodium potassium. And a pump called your sodium potassium pump will bring you back to your resting memory potential at minus 70 millivolts. All right, let's clear all this and go back to the video. Let me back it up a little bit, okay?
Gated sodium channels creation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Okay, so You want to get out of this. What is an action potential? It's a brief reversal of the inside resting memory potential from -70 to positive 30 millivolts. You want to get out of this that if you create an action potential in a muscle, the muscle will contract. Whereas, let's say if you create an action potential in a neuron, the neuron's not going to contract. Well, it's going to release neurotransmitters. And vice versa, let's say we create an action potential in a gland's going to release what it makes in response to an action potential. Okay, let's back it up and let's look at the events at a neuromuscular junction. Now, a neuromuscular junction is a point of communication between a neuron, which is a nerve cell, and a muscle cell. And let's see, I'm trying to magnify this here.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron.
Okay, notice they said an action potential propagates down the motor neuron. What they mean is voltage-gated channels. When you open up one, they all open along the axon, creating your action potential at the end of the tail, causing the release of neurotransmitters. So let me see if I can do the pen scenario. So remember, the first type of channels that open up in response are called chemically gated channels for sodium. Once those open, positive ions for sodium come in, and if I reach a threshold about -50 or -55 millivolts, the next type of channels that will open up for sodium are chemically gated. And the chemically gated ones are located starting at this area of the tail, the axon, called the axon hillock. When one opens, they all open, propagating, which means spreading, opening all along the axon, which will eventually cause the release of neurotransmitters. Okay, let's kind of back it up a little.
An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps.
All right, let me just bring to your attention, before you see the release of the neurotransmitter acetylcholine, not only do you need calcium for a muscle to contract, we need calcium even for the neuron to release its neurotransmitter. So somewhere along here, voltage-gated channels for sodium are going to open, and then you're going to have a voltage-gated channel for calcium open. And calcium is going to come in from outside the neuron, not inside, but outside. And that's going to help cause to release neurotransmitters to communicate to the muscle cell. Okay.
At the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
All right, let me jump in real quick. These are the chemically gated channels for sodium. So once acetylcholine binds to it, it's going to open. Sodium is going to come in. Okay, you're going to see a little bit of potassium leave, but this channel really allows more sodium to come in. So as more positive ions come inside the muscle, it's becoming depolarized. Okay, and when we reach a certain threshold, you're going to see it in the video. You're going to see that voltage gated channels will open, and then that'll create your action potential in the muscle cell. They're not going to go in detail. They're going to just quickly, briefly go over it. But anyway, let me continue.
Step 5. These ligand-gated cation channels open.
They're chemically gated channels for sodium. Another word for chemical gated, you could say ligand gated. Ligand is just a chemical.
Channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so those are the steps between the communication of a neuron and the muscle cell. They didn't go in detail yet in the muscle contraction. We'll get to that in a little bit. Let me exit. Let me go back. Now, let's pick up from where the muscle got excited. We're going to see the excitation spread down the cell membrane of the muscle cell called the sarcolemma. And let's remember that the calcium, in order to be released, you need to create an action potential. And the calcium for a muscle to contract, specifically skeletal, is located in an organelle called the sarcoplasmic reticulum. So we're looking inside a muscle cell here, and what you're looking at in blue is the sarcoplasmic reticulum. It's the organelle that houses calcium inside the muscle cell. And this region of the sarcoplasmic reticulum is called a terminal cisterna. So you got two terminal cisternas, and in the middle, you got an infolding of the cell membrane called the transverse tubule. So these three components, they make up what's referred to as a triad, referring to three. They are going to mention that in the video, but I just thought I'd bring it to your attention before I start the video. And I'll try to be quiet in this video. Let's go ahead and start it.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
All right, so far you saw the release of neurotransmitter, and that causes the release of calcium. Eventually, when creating action potential in the muscle, causes the calcium to be released. Then the next video, we're going to look at how the calcium is exactly involved in the action potential. And let's get to that video. When myosin, that protein called myosin, when it binds to actin, it's called a cross bridge.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, these are animations to help you kind of visualize the muscle contracting and how it's being communicated to you through a neuron. And let's see, where can you find these? They're located within your canvas. You click off of it. If you go to modules, under modules, you can click where it says study area. Within the study area, continue new window, of course. Let's get to it. Go to, let's see here, an animation videos, A&P Flicks. And it's the top one where the videos that I was using are located. Okay. Now, let me go ahead and get back to the PowerPoint. Just give me 1 minute to get to it. Okay, let's just kind of review real quick. When you look at the muscle from large to small, okay, first of all, muscles are attached to your bones. If they're attached like in a rope-like connection, that's going to refer to as a tendon. So I'm just getting the marker out here so I can highlight things. So here's our tendon. Now, the outer connective tissue surrounding the whole muscle, again, is referred to as the epimysium. The units inside the whole muscle, if I were going to pull one of these units out here, that's going to be called a fascicle. And they went ahead and they magnified it for us here. And the outer connective tissue around that fascicle is referred to as perimysium. Peri means to surround. And then the units within the fascicle, okay, let's pull one of these out. Then you're at the cell level, and fancy name for the cell is muscle fiber, or you could say muscle cell. The fiber is just indicating that it's very long. And then the connective tissue around the muscle fiber is referred to as endomysium. Okay, I'm gonna clear all that. Let's go to the next slide. So again, we're looking inside the muscle cell here at a microscope level. You're going to see features called striations where you're going to see dark regions. The dark regions are referred to as the A band, and then the lighter regions are going to be referred to as the I band. Okay. And skeletal muscle, you'll have many nuclei per cell because developing as an embryo, you had single cells fused into one long muscle cell. So looking at the drawing here of the muscle cell, the connective tissue, if you recollect, it's not drawn here, be referred to as endomysium. However, the cell membrane of a muscle cell is called sarcolemma. And the units, to pull one of these units out of the muscle cell, it's called a myofibril. And you're going to see striations again, these stripe appearances. If it's a darker striation, in general, it's called an A band. If it looks lighter, it's referred to as an I band. Now we're looking at a myofibril. Let's have a look at this. What causes these striations are the proteins inside the myofibril. Okay, you got a protein that's thicker in this reddish brown coloration here. It's referred and called as myosin. And then you have a thin one drawn here in blue. The thin one is referred to as actin. And it just, to me, makes sense. If they overlap, right? They overlap each other, it's going to look darker. So that would be our A band. And where you only see the actin, it's going to look lighter. It's going to be the I band. So what causes the striation appearance are the arrangements of these proteins, myosin and actin. And again, the functional unit is from one Z disc to another Z disc. And again, the functional unit is the smallest component of the system here that does its job, which is to get shorter in length, to contract. It's an area from one Z disc to another Z disc. It's called a sarcomere. Now, this is showing us the organelle that surrounds your myofibrils. It's called the sarcoplasmic reticulum. Okay, it's an abbreviated SR. And you got two sides of this sarcoplasmic reticulum where calcium can be released because calcium's stored inside the sarcoplasmic reticulum. These two sides of the SR are called terminal cisternae. And in the middle is an infolding of the cell membrane, the sarcolemma. So this infolding of the sarcolemma is called a transverse tubule. So it makes sense. Two sides, 1, 2 of the terminal cisterna, and down the middle, the transverse tubule make what's called a triad, referring to 3. Okay, let me skip that. So here you're looking at a sarcomere. You're looking at the myosin in brownish red. Myosins, they have these little head regions where they like to bind to actin. So you see little heads here. They like to bind to the protein right above it called actin. All right, let's get to a larger picture. So in this drawing here, you could see that myosin, this protein here, will bind to actin. So there's some binding sites here for actin. There's also a binding site for ATP. And we need ATP in order to move this hinge. This is a hinge. Think of your elbow like you have a hinge joint in the elbow. So now we're looking at actin. Right now, the binding sites where myosin would like to bind to, they're covered up by a protein referred to as tropomyosin. And in order to expose those binding sites, I need to release calcium. Calcium that's stored in the sarcoplasmic reticulum. The only way we're going to get calcium release is to create an action potential inside the muscle. In other words, we need to change the voltage inside from minus 70 millivolts to positive 30 millivolts. Once calcium's released, it'll bind to a protein called troponin. That'll push aside the tropomyosin, exposing the binding sites on actin, like we saw in the video. All right, so let me get to the picture here. And when myosin binds to actin, there's ATP. The actin on both sides of the Z disc here are going to move toward the middle, and the sarcomere gets shorter in length. That's referred to as a sliding filament theory. All right, so going back to the neuron, neuron's a fancy name for the nerve cell. We're going to see that the neuron is excited, and it's going to cause the release of neurotransmitters called acetylcholine. Okay, the end of the axon, this round region here, it's called the axon terminal. It's labeled right here, axon terminal. So we do need calcium, not just for the muscle to contract. We saw in the videos, you need calcium to guide these little vesicles called synaptic vesicles that contain the neurotransmitter, guide them to fuse their membrane into the axon terminal. And that will cause the release of the acetylcholine. Let's see if we get a larger picture. No, we're not. So I'm going to stick with this picture here. Once the acetylcholine is released, it's going to bind to chemically gated channels for sodium. So acetylcholine fits perfect on an area called the receptor region on that channel. And it fits perfect like a key to a lock. Otherwise, the channel is going to stay closed. So when the channel opens, it's going to allow for sodium to come in. Okay. Let's go and draw our little graph again. The resting membrane potential is about minus 70 millivolts. Okay. So as positive ions of sodium come in, because the channel is open, it wants to go from high to low, positive ions for sodium come in, the inside is becoming less negative, and the term is called depolarization. Okay, now if I reach voltage, like if I reach a certain threshold, I can open up voltage-gated channels for sodium, somewhere about minus 50 millivolts. If I could change the inside from minus 70 to minus 50 millivolts, the next type of channels that open up on the sarcolemma are voltage-gated channels for sodium. That means they're only going to open up when they sense it's about minus 50 millivolts. When one voltage-gated channel opens, they'll all open, creating your action potential, which means you went from minus 70 to positive 30 millivolts. and that will cause the release of calcium from the sarcoplasmic reticulum. All right, so let me jump forward. So here it is showing us that one voltage gated channel opens. They should all open. So this is a voltage gated channel. It opens. The one next to it will open. The one next to that one will open. And the excitation, the opening up of these channels will go down the sarcolemma, which is the cell membrane of a muscle cell. So here I got voltage gated channels open, similar to dominoes. If you hit one domino, they're all going to fall. So if I open up one voltage gated channel, they all open. So as they open, you're changing the inside everywhere it opens to minus 30 or positive 30 millivolts, creating your action potential. I know that's bad. But anyway, once I create my action potential, that causes the release of calcium from the sarcoplasmic reticulum. Once the calcium is released from the sarcoplasmic reticulum, and it's actually released in an area called the terminal cisterna. Remember, you have two terminal cisternas of the sarcoplasmic reticulum. Okay, so calcium's released, and we need the calcium in order to expose the binding sites on the actin. So right here is the protein called actin. The binding sites are covered up by a protein called tropomyosin. However, when the calcium is present, it binds to troponin. And once the calcium binds to the troponin, that'll push aside the tropomyosin, and then they'll expose the binding sites on actin. And as long as those binding sites are exposed, the myosin protein here can bind to the actin. And when myosin binds to actin, we form what's called a cross bridge. The only thing we need now in order for the muscle to get shorter in length is ATP. So this is the kind of steps how it works. Let me start off here, okay? So previously, before ATP was present, you had a cross bridge. You had a myosin bound to actin, okay? I'm drawing myosin here. But when ATP is present, it disattaches the myosin. So ATP is needed to disattach myosin. You probably heard of rigor mortis, where after death occurs, the body kind of gets like in a stiff state. And what causes that is you're no longer making what? ATP. So any cross bridges where myosin's bound to actin, it's not going to come off until time, natural decay occurs. So when you no longer make ATP, you pass on, you'll see rigor mortis occur. Okay, so ATP is present. That disattaches the myosin from actin. In other words, it disattaches the cross bridge. What's next? Well, let me back it up a little bit. ATP is going to get hydrolyzed. Hydro means you're going to add water and separate it into ADP and phosphate. So you previously had ATP. ATP stands for adenosine triphosphate, which means you have three phosphates attached to adenosine. Now, if I take one off, I'm left with only two. So a phosphate's being taken off. So ATP gets hydrolyzed into ADP and phosphate. Okay, so what? What did that do? When that hydrolyzes ATP, that allows for the hinge to be pulled back. So that way it can get ready to contract. Once the ADP and phosphate leave, you're going to see the myosin move actin, and that's called the power stroke. Let's go to the next slide. So here you see the ADP and phosphate leave, and that will now provide the energy to move actin, which is called the power stroke. And if you can remember, the myosins on this side, they're moving actin toward the middle from left to right. And the myosins on their hinges on this side are moving the actin from right to left, so that the sarcomere from one Z disc to another Z disc is getting shorter in length. And get enough of these sarcomeres to do that all together, the whole muscle will get shorter in length. Let's clear all these drawings out. So there the muscle contracts. Okay, great. Now we need to come back to rest. The muscle has to return the inside to minus 70 millivolts, going back to our resting memory potential. All right, so let's kind of review here. So step one, a neuron, and let's just say we're at minus 70 millivolts, not minus 90, okay? Step one, a neuron releases neurotransmitters onto the muscle, opening up chemically gated channels. As chemically gated channels open, positive ions for sodium come in, the muscle's becoming depolarized. When you reach a certain threshold, in other words, when you change the voltage to about minus 50 millivolts, the next type of channels for sodium that will open up are referred to as voltage gated. Once you open up the voltage gated channels, they all open, similar to dominoes falling. And that creates your action potential, which is the reversal of the inside from minus 70 millivolts to positive 30 millivolts. So at this point, the muscle's contracting, okay? You got calcium released, myosin's bound to actin, the muscle's contracting, okay? Now at 30 millivolts, all sodium channels should now be closed. All sodium channels are now closed. So let me back this up now. After all sodium channels are closed, we want to come back to rest. The next type of channels that are open up in order to come back to rest are voltage gated, but for potassium. So the voltage gated channels for potassium are only going to open up when it's already 30 millivolts. You create your action potential, you're at 30 millivolts, that'll cause this channel to open, and because there's more potassium, it wants to leave, just like in the picture here. So as potassium's leaving, you're losing positive ions. All right, let me get back to the graph. So let me draw my little picture here. So as we're losing potassium now, because voltage gated channels for potassium are open, you're losing positive ions. You're now coming back to rest from 30 millivolts, coming back toward minus 70 millivolts. The term is called repolarization. Now these channels, they're open up a little longer than you need. You do go a little bit below minus 70 millivolts, called hyperpolarization. Finally, they will close, and your leaky channels, which are channels just naturally always open, and your sodium potassium pump will bring you back to minus 70 millivolts. Okay. And I think we will pick up and we'll cut this video into two sections where we covered the first, the anatomy and the steps of an action potential. And then the next video I'll finish up this chapter and post it hopefully somewhere within this week and let y'all kind of absorb it a little at a time. And I'll see you next time.